For our symposium, we chose to research T-VEC oncolytic viruses and how they can be used to fight various cancers. Oncolytic viruses are viruses that have the ability to target and kill tumor cells while protecting healthy cells. Today, cancer treatments struggle to kill tumor cells without harming healthy ones, which can be harmful to patients' immune systems and blood cell count. Oncolytic viruses leverage the natural ability of viruses to infect and replace within the body's cells and expose their vulnerabilities inherent to cancerous tissues. This highlights the molecular features of malignant cells and distinguishes them from healthy ones.
Scientists have found a way to engineer these viruses to enhance their naturally occurring ability to inherit cancer cells selectively. Oncolytic viruses can cause great destruction to the body's mechanisms, including direct lysis and the induction of apoptosis. The targeted approach of oncolytic viruses minimizes the damage to surrounding healthy tissue. This also triggers an immune response that alerts the body's immune system of cancer cells' presence. Furthermore, it enhances the immune system's ability to recognize and attack cancer cells.
Our research is specific to T-VEC, the first oncolytic viral immunotherapy approved for the local treatment of unresectable metastatic state 3BC IVM1A melanoma. T-VEC is a genetically modified herpes simplex virus type 1 designed for oncolytic purposes. This virus has been altered by scientists to replicate selectively within tumor cells, which leads to the tumor's cell destruction. T-VEC also carries an inserted GM-CSF factor gene, which enhances the anti-tumor immune response.
The study we chose to research was designed as a clinical trial that looked into the effectiveness of T-VEC in treating superficially accessible melanoma. The study included 436 patients with stage 3 and 4 melanoma that were randomly assigned in a 2-to-1 ratio to receive either T-VEC or recombinant granulocyte macrophage colony stimulating factor, or GM-CSF. The results showed an objective response rate of 26.4% for the T-VEC in comparison to 5.6% for the GM-CSF, and durable response rate of 16.3% for T-VEC compared to 2.1%. These results met the primary endpoint of the study.
In addition to the promising results, T-VEC patients exhibited an overall improved survival compared to the GM-CSF therapy. The overall survival improvement of T-VEC compared to GM-CSF consistently showed a 5-year survival rate of 33.4%. With such positive outcomes, the study confirmed the safety of T-VEC for melanoma patients and received approval from the U.S. FDA in 2015. It was approved for the local treatment of recurrent melanoma after initial surgery.
Due to the promising results found in this trial, we recommend further research of T-VEC in the treatment of other types of histological cancers. Accessible tumors such as head and neck squamous cell carcinoma, soft tissue sarcoma, and breast cancer could also be treated. When first conducting these trials, the primary goal would be the safety of administration of T-VEC instead of the actual effects on the cancer. Once safety is controlled and observed, testing may begin.
One example is a trial done where 17 participants who had squamous cell carcinoma received T-VEC combination with other cancer treatments. This shown after 29 months a survival improvement. Disease-specific survival was seen at 82.4%, and although this percentage is small, the results support further studies to be done in the neck and head cancer.
After clinical trials were conducted, patients were more responsive to the virus and saw an impact on their survival when undergoing treatment. With this advancement came numerous challenges faced when testing.
The dosage of T-VEC must be carefully monitored since it is different for the initial injection versus its later ones. This is because the volume required for the virus for a patient can only be determined when the patient has a tumor measured, which can cause delays in ordering and wait times for patients. A challenge faced when injecting the treatment is it requires direct access to a patient's tumor site and the manual administration of a physician. Tumors for some patients may not be palpable or may be a size undetectable, making it hard for injection.
Further challenges arise with biosafety concerns. Oncolytic viruses are typically live, which would require clinics and pharmacies to enforce biosafety measures for spills and waste. With the use of this treatment does come the risk of viral transmission by close contact. To prevent transmission, it is important to educate patients to prevent direct contact between patients' injected sites and other individuals. This includes educating other healthcare professionals as well.
Our group also faced some challenges during our research process of this advancement. such as understanding the information we were analyzing and working as a cohesive and effective team. Most of our struggles could most likely be aligned with the research team of this advancement. The most prevalent would be as working as a team effectively. In order to reach an end product, the research team must have managed their time and communicated with one another cohesively. This translates to how our group project also required the same.
Thank you for listening to our research study for using T-VEC oncolytic viruses to fight certain cancers and our proposal to further study on head and neck cancers. And a huge thank you to our AP Bio teacher, Mrs. White, for sponsoring our research proposal.